of competitors, such as the mammalian radiation that occurred after the KP mass extinction event. There's the appearance of new resources in an area, such as the appearance of grasslands in North America. And there's the appearance of a key innovation, which allows a lineage to interact with previously unavailable resources. Focusing on ecological opportunity through colonization, most studies focus on isolated systems such as islands, as it provides a clean case study with an obvious barrier. However, when it comes to a continental system such as North America, the complexity of different environments and the transitions between those environments could provide a similar level of isolation for certain lineages, thus providing ecological opportunity for lineages that are able to cross these barriers. This is the snake in the snake tribe Thamnophini. So if you're familiar with North American snakes in any way, you should be familiar with these fellers. They consist of perhaps the most familiar and abundant snakes in North America. So a quick roll call, you have your garter snakes, your water snakes, your brown snakes, and of course, you got your swamp snakes. They are incredibly ecologically diverse and occupy numerous different habitats and have a wide array of diets. And you see this asymmetric diversity at the generic level, where most genera have anywhere from a single to 10 different species. But the genus Thamnophis, otherwise known as the garter snakes, have 38 different species, consisting of about 2/3 of the diversity of the clade. And you see this asymmetry presented in a geographic context, whereas all other genera are confined to the eastern Arctic. Thamnophis lineages are able to disperse westward in 2 different instances. As you can see here, broad-scale geographic patterns in the clade are primarily structured along the Western Continental Divide, which is defined primarily by a transition from mesic and low elevation environments in the east to xeric and high elevation environments in the west. Now, the ability of Thamnophis lineages to cross the Western Continental Divide and persist provides ecological opportunity, which is reflected in its phenotypic evolution. There are numerous instances of convergent evolution between Eastern and Western taxa. And there's evidence of state-dependent phenotypic evolution being higher in Western lineages than Eastern lineages, supporting the hypothesis that Thamnophis lineages are taking advantage of ecological opportunity when they cross the Western Continental Divide. So we have evidence that ecological opportunity in the West is driving phenotypic evolution in Thamnophini. However, it is unclear what are the underlying mechanisms that allow Western lineages to cross the Western Continental Divide and take advantage of ecological opportunity, particularly at the genotypic level. However, due to advances in whole genome sequencing technology and bioinformatics, I can identify entire regions of the genome that help facilitate ecological opportunity. And that is the focus of this talk. So my objectives here are simple. First, identify which genes are being positively selected in Western taxa. Compared to Eastern taxa, I expect Western taxa to have a higher proportion of positively selected genes to rapidly evolve into new Western environments. And second, determine if genes positively selected in Western taxa are associated with environmental variables that define the transition across the Western Continental Divide. If these genes facilitate dispersal across the Western Continental Divide, thus allowing Western lineages to take advantage of ecological opportunity, it stands to reason that these genes are being positively selected to adapt to environments specific to the Western Continental Divide and the West. So I expect to see correlation with these genes and Western environments. To approach these questions, I utilize whole genome data and a comparative genomics pipeline, which encompasses whole genome sequencing and assembly, whole genome alignment, identification of orthologs, inferring positive selection, and determining correlation between genes and environment. So going through this pipeline, let's first identify which genes are being positively selected in Western taxa. So for this study, I assembled the largest genomic dataset yet for Thamnophini of 61 short-read whole genomes. This encompasses 90% of total Thamnophini diversity. Genomic coverage ranges from 20x to 60x coverage. And for reference mapping and annotation transfer, I use the Thamnophis elegans reference genome from the Vertebrate Genomes Project. And a whole genome alignment was used in downstream analyses, was generated using progressive cactus. And downstream comparative phylogenetic analyses use a phylogeny of Thamnophini that I inferred in a previous study. For selection inference, I conducted adaptive branch site random effects likelihood tests on protein coding regions of the genome. Abstral tests if positive selection has occurred on a proportion of branches along the phylogeny. So I ran abstral in 2 different modes. First, in an exploratory full phylogeny mode where all branches are tested for selection to determine if there are a greater number of genes under positive selection in Western taxa than Eastern taxa. And second, I tested for positive selection in Western taxa only as a set of foreground branches to more powerfully determine which genes are under positive selection in Western taxa. So looking at the results, you can see here that there's a far greater proportion of genes under positive selection in Western taxa than in Eastern taxa. It becomes clearer when mapped onto the phylogeny we see bursts of positive selection of genes within the western clades of Thamnophene. Now, when testing for positive selection explicitly in western taxa, I found that approximately 59% of all tested genes were significant for positive selection in western taxa. So I was able to identify these suite of genes that are undergoing positive selection in western taxa, but is there really a relationship between these genes and westward dispersal? So let's move on to the second objective, which is to determine if these positively selected genes are associated with east to west dispersal. Specifically, are genes under positive selection in western taxa associated with environmental variables that define a transition across the western continental divide? Again, it stands to reason that genes undergoing positive selection in western taxa are doing so to adapt to these shifts in environmental variables across the western continental divide. However, to test for this, it is important to determine which environmental variables define this transition specifically for Thamnophene. To do so, I ran this comparative phylogenetic pipeline to identify which environmental variables have adaptive regimes that overlap with western clades, again using my previously inferred phylogeny. To collect environmental data, I utilize a modified version of a recently published pipeline called Candy, which gathers and cleans occurrence data, downloads environmental data, and estimates environmental niches for numerous taxa at a time. Environmental niche modeling was done using Maxent, using world-class bioclimatic variables and Earth and topographic variables. And models were fine-tuned, removing correlated variables in the final models. The best fitted model was chosen for each species, and environmental variables were extracted from localities that represented the highest probability of occurrence from each niche reconstruction. The idea here is that these variables represent the optimal value for that variable for that species. And to identify regimes for environmental variables, I used Bayou, which models the evolution of a continuous character under an Ornstein-Uhlenbeck process. This allows for the optimum of the OU process to vary along the branches of the phylogeny and identify placement of those regimes along the phylogeny. And I ran these analyses across all environmental variables. Again, the idea here is to identify which environmental variables have regimes that overlap with western clades. To give an example, here is one of the variables where adaptive regimes do overlap with western clades: precipitation of the driest quarter. As you can see from the trait map on the left, there's a clear delineation of the optimal values of precipitation of the driest quarter between eastern and western taxa. And you also see this represented in the phylogeny here, where the different adaptive regimes are color-coded. And you can see that they largely overlap with western clades. So from the Bayou analyses, I identified 8 environmental variables where you see a similar pattern of adaptive regimes overlapping with western clades. These 8 variables include macroclimatic, microclimatic, and topographic variables. And to test correlation with positively selected genes and environmental variables, I use RER Converge, which is an R package that tests for the correlation between the relative evolutionary rates of a set of genes and the evolution of a trait across a phylogeny. Since we already know that the genes tested are under positive selection, I expect to see genes that are correlated with environmental variables to have accelerated rates of change compared with environmental variables, indicating that the genes are under positive selection as a response to adaptation to these new environments. So based on the RER Converge analyses, I found over 1,000 genes that are correlated with these environmental variables that also exhibit accelerated rates of change, in particular in response to isothermality, relative humidity, and topographical roughness, indicating that these genes are undergoing positive selection in response to adaptation to these environments, thus allowing western lineages to cross the western continental divide and persist, and thus take full advantage of ecological opportunity. Now, with that, what are the key takeaways from this study as it relates to the broader story of diversification of Thamnophene? Well, first, I found out that a greater proportion of genes are undergoing positive selection in western taxa when compared to eastern taxa. And over 59% of genes that were tested were undergoing positive selection in western taxa. And I also found out that out of those positively selected genes, over 1,000 of them are correlated with environmental variables that define a transition across the continental divide and undergo accelerated rates of molecular evolution, indicating that these genes are undergoing positive selection to adapt to these new environments. So to put this into the broader context about diversification in Thamnophene, you see that there is an asymmetric diversity in the group in both the species and geographic context, driven by Thamnophis lineages crossing the western continental divide and taking advantage of ecological opportunity. In this study, I identified the suite of genes that facilitates the crossing of the western continental divide for western lineages in Thamnophene, thus allowing these lineages to persist in the west, take advantage of ecological opportunity, thus leading to greater phenotypic and species divergence. as well as showing how radiation can come about in a continental system. Now, what is next is to explicitly tie this to function. Now, as you can see from this preliminary gene enrichment analysis of positively selected genes, these genes are associated with a wide variety of physiological and cellular mechanisms. However, incorporating actual physiological and gene expression data will even further clarify the function of positively selected genes and the underlying physiological and molecular mechanisms that facilitate ecological opportunity. And to do that, we require collecting more genomic data, particularly transcriptomic and gene expression data that can clarify the functional aspect of positively selected genes, as well as population level data to better infer intraspecific variation in these genes, particularly for wide-ranging taxa. It will also be incredibly useful to collect more trait data, particularly physiological and behavioral data, to better understand how Western lineages are responding to changing environments and how it relates to the genome. And finally, it will be important to test for correlation between morphology and genomics, particularly to see which genes are driving convergence between Eastern and Western taxa. And with that, I would like to thank all the people listed here, in particular, the collection staff at the various natural history museums that I heavily relied on. And with that, I'll take any questions.
Thank you very much. We have a few minutes for questions. People can put them in the Q&A or in the chat, or if you raise your hand, we can unmute you. So ask by your preferred means.
I don't know if I can see the chat.
We can see the chat so I'll start off with one.
Yes.
It seems like, so you had all those snakes that went east to west, but it looked like, were there a few that went back east? Can you look at whether the opposite selection
Yes, there's one clade in particular, garter snakes. It's this clade, it's an interesting clade of snakes that are in the Midwest. So it's like Radix, Brachystoma, butleri, these three species that, based on ancestral geographic reconstruction, we found that the ancestor for that lineage originated in the West, and then they moved back east around the same time that grasslands were popping up around in the Great Plains in Central North America. As far as I can tell, there didn't seem to be any obvious breeder selection in those species. Just looking at the full phylogeny actual selection test, as far as I understand, but I'm sure looking at further analyses and looking at those at that clade in particular, I'm sure that there'll be some interesting things happening there. So, yeah.
Thanks. We've got time for one more question. Well, Benjamin, if you go ahead and start your slides.
Looks like there's a question in the Q&A.
Okay.
Are you able to see that, Leroy? I can read it.
I can see it.
Okay.
All right. That was a really cool talk. Some of the estimates for genes under selection are wild. Do you think for some of these species, truly 20% of the genes are under selection, or are many of these potentially looking like they are under selection either due to hitchhiking with selected sites, or maybe impacts of bottlenecks on those species, making things look like selection? That's a great question. So when I looked at all the different species that I tested, so only a couple.
Sorry, we're actually, sorry to break in, but we're actually going to need to move on. So if you could just engage over the chat.
Yeah, you can type your answers in there.
Okay, I'll do that. All right.
And our next talk is by Ben Tao. You need to unmute yourself.
Okay. Okay, so hi. Today I'll be talking about how tapping into the graphical models literature can benefit both practitioners and methods developers studying evolution on networks. And this is joint work with my excellent collaborators, Paul and Cecile. Okay, so our starting point is the phylogeny for a set of species. So if you look at the top figure, this could be a tree as shown on the left or a network as shown on the right. The nodes in the phylogeny represent species and the edges represent descent. But in the case of a network, we can have hybrid nodes. So for example, here where we have multiple parents, and this is useful for explicitly modeling gene flow events such as at mixture, migration, introgression, hybridization, horizontal gene transfer, recombination, etc. So I'm interested in modeling trait evolution on a network, but unfortunately, evaluating evolutionary models on larger networks tends to be disproportionately more costly. So for example, if you look at the bottom figure, this shows some characteristics of empirical and simulated networks. And the x-axis measures network complexity, and the y-axis measures the cost of model fitting associated with network complexity. So for example, sure, you could increase the size of the network, but if inherently the network has a fixed level of complexity, then the maximum cluster size won't increase. So another thing to note is in this plot, the darker colors indicate larger networks. And the message this is trying to say is that larger networks tend to be more complicated, and more complicated networks are more costly for model fitting. So as an example, suppose we wanted to model how virulence evolves for some pathogen. So if we have a tree with 40 tips, then evaluating a model on this phylogeny is not that costly because trees have low complexity. So the maximum cluster size is 2. But recombination and horizontal gene transfer are known to be prevalent in pathogens. And so the true phylogeny is probably quite reticulated. And thus the cost of evaluating a model on a reconstructed network can be high. So to facilitate larger studies that can account for reticulate evolution, we do need more efficient methods for evaluating models on networks. And the plug I'm making is that studies and methods that scale are the future. So just an overview of some of the technical details. So why is model fitting on a network costly? Well, our model basically tells us how to compute the probability of observing particular trait values at all of the nodes, but we can only really observe the tip values. And so what we really want to do is compute the probability of these observations alone. So this is what we call the likelihood, and it's basically how we score different parameter values for our model. And to find good parameter values, we may have to calculate this score for different candidate parameter values multiple times. But performing the summation involved to calculate this is costly, and this is especially so for the case where the ancestral relationships are complex. Okay, so some general strategies we can take to address this computational bottleneck are to perform the summation cleverly, to compute an approximation that's maybe less costly, and also to compute the likelihood gradient, so to tell us how to search for better parameter values more efficiently. And it turns out that all of these approaches are facilitated by the graphical models framework, which I'll now give an overview of. So in a nutshell, the graphical models framework basically starts with identifying the direct dependencies among the variables we're modeling. So for example, in the case of vertical descent, the trait of a species depends directly on that of its parent, and this relationship could, for example, be modeled as a single edge. And in the case of gene flow, the trait of a species may depend on multiple parents, and this relationship can be modeled as a hybrid family consisting of multiple edges into a child node. And so we can represent these dependencies jointly by a graph, and then we can apply generic algorithms that process groups of these nodes in the graph to build up various quantities of interest. So again, the quantities of interest could be a likelihood, an approximation to it, or the gradient. And so actually a new contribution that we made was finding a way to leverage these algorithms to build up the optimal parameter values for the homogeneous Brownian motion model. Okay, so the graphical models approach is not exactly entirely new to the evolution community. So for those of you who are more familiar with algorithms in phylogenetics, you will know the pruning algorithm. And the pruning algorithm is basically a special case of this graphical models framework when the graph of dependencies, I'm just going to refer to this as G, or the graphical model, when this G is a tree. So what happens is we process sister nodes in post order, and by the time we reach the root, we've accumulated the likelihood. But for the more general case where G is a network, or G is permitted to have these hybrid nodes or these cycles, then what we have to do is build an intermediate graph structure called a cluster graph, and then we process its clusters in post order. So the cluster graph is just another graph structure where the nodes now consist of groups of nodes from the original graphical model. So the algorithm is known as belief propagation. And I talked about how we could build up the exact likelihood, and also we could build up the approximation. And how we do this is by customizing the cluster graph. So for example, if the cluster graph we built is a tree, then by running this algorithm, we accumulate the exact likelihood, but we can also introduce loops. And when the cluster graph is loopy, then what we accumulate is an approximation. And so a natural question is to ask, well, why would we have the approximation? Well, it turns out that when we introduce loops, we can have smaller clusters, and smaller clusters allow us to do the computation less costly. So it's a trade-off between approximation error and scalability. Okay, so another natural question is, well, how do we find the graph of dependencies? How do we find this G? Is G always the same as the actual phylogeny? And the answer for that is that for simpler models and for Gaussian models, it typically is. But for more complicated processes, it may not be. So if you consider the evolution of a biallelic marker, and for example, we observe one or more individuals from each species, and we want to account for incomplete lineage sorting, then the situation isn't that simple. So for example, if we track the number of red alleles as well as the total number of alleles within each population as we go backwards in time, then we can see that the graphical model actually is quite a bit more involved than the actual phylogeny. So here I'm just showing you a speciation event, and I'm drawing the graphical model for the variables involved in this speciation event, and you can see it's quite more complicated. So the key message is that G depends both on the network and the model. Okay, so far I've talked about an abstract framework, but what about concrete steps towards an implementation? So, well, I'm currently working on software that applies this framework to the continuous trait setting. And so continuous traits are widely studied. Examples include morphometric data, gene expression, allele frequency, and pathogen virulence. And what our current prototype does is it models continuous trait evolution using linear Gaussian models. So this is the case where the child state depends linearly on its parents plus some Gaussian noise. And this basically includes most, if not all, of the Gaussian trait models you may be familiar with. And so here I'm just showing you a well-known case of Brownian motion along an edge where the state of the child is basically the state of the parent, plus some Gaussian noise where the amount of variance depends on the length of the branch and the evolutionary variance rate. Okay, so I'm working on the software, and so here we're demonstrating our prototype on leaflet data from the genus Polemonium. So this is a flowering plant genus with compound leaves, and it's widespread in North America and Northern Eurasia, with both broad latitudinal and elevational range. And the key things are that its leaflet size varies greatly across species, and we also know that gene flow occurred. So here we use the software to fit a Brownian motion model of evolution for leaflet length along this 17 taxon network. And the table below presents the model estimates and likelihood for 3 available options. So the first row is showing the results of our new approach, which builds up the optimal estimates directly. And the second and third rows show the results when we try to maximize either the exact or the approximate likelihood by iteratively trying out different parameter values. So in this example, the network is too small and simple to accentuate the scalability gains of the approximate approach, but it does show that the approximate approach can produce good estimates. Okay, so to recap, networks are needed to model reticulate evolution more accurately. Efficient and scalable methods that apply to larger, more complex networks are needed. the graphical models literature offers a flexible framework and rich tool set to tap into. And lastly, we demonstrated proof of concept for how this approach can be applied to the continuous trait setting. So there's still more to be done for both practitioners and methods developers. So on the application side, as network methods improve, large-scale studies that better account for reticulate evolution can be carried out. And on the method side, although the graphical models literature is extensive, the pattern of data in traditional applications, such as engineering, is somewhat different from the pattern of data in phylogenetics. And so, in my view, there is plenty of room to adapt and specialize these methods to our purposes. Okay, so here are some of the references, and thank you for your attention.
Thank you. And again, we have a few minutes for questions. There's one in the Q&A already from Brian O'Mara. And I don't know if you want to read it or if we should read it. He's saying, it seems that the approximation only approximates in the case of cycles and that the tree part isn't approximated. Is there a bias where the cycles are less likely to be inferred correctly under approximation?
I think what I meant was that as long as your cluster graph has cycles, then you will produce an approximate likelihood. And as long as your cluster graph is a tree, then you will produce the exact likelihood. So you could have a network that has cycles, and if you build a cluster graph that's a tree, then you will not have approximation. I'm not sure if that answers your question.
Brian says thanks in the chat. Got time for one more question? So I have a quick one, which is, I'm assuming that this is carrying over all of the usual assumptions in terms of how the continuous traits are evolving.
Right. So so I guess the short answer is that typically our models of evolution assume that evolution along different branches is independent. But when you introduce more complicated things such as the coalescent, then that's not exactly true, and that's why we have this graph of dependencies, this G, which is not exactly the same as the phylogeny itself. Yeah.
Okay, thank you very much. We'll take a 2-second breather and then we'll be on to the third talk of this session, which is by Sarah Morris. Sarah, it's all yours.
Okay, let me just go into present mode. Okay, can everyone see that all right?
Yep.
Okay, so then I am going to be talking about what I'm calling the fishing spider conundrum, paraphyly, and the restoration of the family Dolomididae. So to begin talking about Dolomididae, we need to talk about Pusaridae, because this is the family that those genera currently belong to. So Pusaridae is known for having these nursery webs. They're called nursery web spiders, and that's because they all create these webs for their young where they enclose them and then let them out later when they're older. And this isn't actually a synapomorphy of the group, though. Nursery webs are made by other spider families like Trachelidae and Ctenidae. Fusariids are also unique because some of them are known as fishing spiders, where they are semi-aquatic, and some of them will walk along the surface of the water to catch aquatic prey. Some of them can even dive to evade predators. It's really cool. But fusariids have a variety of lifestyles, so not all of them fish. Some of them build catching webs, some of them wander and ambush prey, and so on. So to give you an idea of where these spiders lie in the tree of life, they belong to the superfamily Lycosoidea, and these are wolf-like spiders. So they include the wolf spiders, of course, like this very charismatic Hogna species up here. Wolf spiders are some of those that if you go out at night with a flashlight, you'll see their reflective eyes shining back at you. Other lycosoids include the wandering spiders here, like the genus Phenutria, and these are very unique because they are actually some of the most dangerous spiders in the world. There unfortunately are a good number of deaths attributed to them in South America. So those are some related spiders to the Fusariids. So what got me interested in this project to start with is that there isn't a dedicated analysis of intrafamilial relationships for this group, and of recent studies that have included Fusariids, what you'll find is that a lot of them show that the family is not monophyletic. So 3 of the genera that tend to fall outside of Fusariidae in these analyses are Dolomedes, Bradisticus, and Hygropoda. Back all the way in 1898, Simone actually suggested the construction of a family called Dolomidi. And then in 1967, Lettinen did a morphological analysis and suggested that some of these genera belong in a family called Dolomididae. However, Lettinen's claim was rejected by Searwald in 1990 because she found that his character coding was very ambiguous and the differentiation was unsubstantiated. Then later in 2017, we had this big study by Wheeler et al., and they used only a few Fusariids, but they found it monophyletic, and they considered these genera to be part of a subfamily called Dolomidinae. So it begs the question, of the two, Dolomididae or Dolomidinae, which hypothesis would be supported by a robust taxon sampling and a dedicated phylogeny to this group? So my first research question is, are Fusariids monophyletic? So what I did here, I used a total of 61 Fusariids, all that I could get my hands on. I expanded that taxon sampling from 9, which was the most genera used in a previous study, to 16 genera, so almost doubled it. I included those 3 genera that typically fall outside of the family, as well as Nihilus, which fell outside of the family in one of the analyses. So I have all of those accounted for. Some of my loci were obtained through NCBI's GenBank. I used the 8 molecular markers listed there in purple, and I also used UCEs, or ultra-conserved elements. This utilized the Spider 2KV1 probe set, which was actually developed here in the Ormika lab at George Washington University. And this is a really cool probe set because it can gather over 2,000 different UCE loci. So I aligned the sequences in MAFT and did other data processing in Genius, like concatenation. Then I used IQ-tree for maximum likelihood analysis. And finally, I did a weighted astral analysis to account for incomplete lineage sorting of genes. So this is the maximum likelihood analysis that you're seeing here. The astral analysis had the same topology, so I'll just show this one for convenience. What you'll find is that Fusariidae is split cleanly into these 2 groups with high support. This is 100/99 at the node here. And so in this group down here, what we see are those 3 problem genera from before, Dolomedes, Bradisticus, and Hygropoda. And then up here, we have all of the other Fusariids, including Nihilus. So I'm calling this group down here Dolomitidae. So to give you a little intro to these three genera, I figured I would just go through them really quick. The first one is Bradisticus. This is a New Caledonian species. Their common name is mud wallowing spiders because they have this really cryptic way of covering themselves in mud to blend in with their surroundings. Then we have the Hygropoda genera. These are very different. They're known as leaf-running spiders. They actually capture their prey via sheet webs that they build on vegetation. And then we have this worldwide distributed Dolomedes species where the family gets its name. Now these are your fishing spiders that walk along the surface and catch aquatic prey. So that moves me into my second research question here. Do we have any morphological characters that can corroborate the sequence data? So to explain this, I need to give you a little bit of background on spider anatomy here. So this, what looks like a pair of legs in the front here, is actually a pair of accessory appendages known as the pedipalps, or palps for short. All spiders have these. They can be used for all kinds of things like prey manipulation or moving dirt when they're burrowing. But for the males, they are actually secondary reproductive structures. So what they do is they deposit sperm onto a bit of webbing, and then they charge the sperm into the pedipalps, and then the pedipalps are what is inserted into the female during copulation. So as you can see here, the pedipalps can be really complex and interesting. These are all male pedipalps. The female ones are much more simplistic. The exact structure is always species specific, so we can use them for identification. And one thing that they will always have is this embolus, which is most visible here on the Thalmossia species. It's a sclerotized tube. This is what enters all the way into the female, and then the sperm duct opens at the end there. You'll also see that they have a lot of little apophyses or projections. These are all useful for identification. So some synapomorphies that seem to support this family. One is the saddle, which was identified by Searwald in 1990. And that is this little sclerotized plate here in the center. You can see here that it's present on both Dolomedes and Bradisticus. The next one is a bit of a mouthful. It's called the retrolateral tibial apophysis, and specifically the fact that it's bifurcated. We call it an RTA for short. As far as I currently understand, no Pisarids or other Pisarids have a bifurcated one. Then the last thing is this subterminal lateral apophysis, or SLA. Now, to show you what you're looking at here, you have the embolus, which is this tube, and then you have the structure called the fulcrum that follows along the embolus. And then attached at the base of the fulcrum is this little pointed structure, and that's the subterminal lateral apophysis. You can see it on Bradisticus as well as Dolomedes here. However, those last two structures can't be seen in Hygropoda. So what is the deal with that? Well, in the type species here, there are these two structures. One of these might be homologous to the saddle. That is still to be investigated. And these other specimens on the right here, you can't really see a saddle at all or an SLA. It's possible that these were lost in some Hygropoda. Also, more to investigate for the future. So moving into the female anatomy, the main thing to understand here is that they have this plate on the abdomen called the epigenum. And if you were to look in close at it, it's got these copulatory openings where the pedipalps would be inserted. And if you were to dissect this out and look at the back of it, the inside, you would have all of these tubes. So I won't go too far into this, but what you can know here is that we have the copulatory duct where the embolus is inserted, the spermatheca, which store the sperm, and then the fertilization ducts, which are these harder to see structures. So some potential synapomorphies for this group. The first one is the accessory bulb on the copulatory duct. Searwald mentions this in 1989 for Dolomedes, but you can see here it's also present in Hygropoda, at least on this Bradisticus species. Another one is they all have a reduced head of the spermatheca. Now, what that means is in a lot of spiders, the spermatheca sort of coalesces into a terminal bulb, like a round bulb shape. And none of these have that. They just have these tubes that keep on going. And the last one is this long fertilization duct that ends in a flake-like shape, which you can see they all seem to have, even though this one's hard to see. So to conclude my findings here, we believe that an increased taxon sample does not support the monophyly of this group, and that the resurrection of Dolomitidae as a family is necessary. And so far, it seems that this is supported by morphological characters, though I believe there are probably more to find and more analysis to be done in that regard. A little bit of research I'd like to do in the future. These are Australian, New Caledonian, and New Zealand species of Pisarids that seem to have all of the same structures I was talking about before, but no molecular data currently exists for these species. So I would love to find out in the future where those would fall on the phylogeny. The same can be seen here for the female reproductive structures. So that concludes my presentation. I just want to say thank you in particular to Hannah Wood, Tom Nguyen, and Nikolai Sharp for specimen acquisition, the Mortensen Fund at George Washington for funding, and my 5 friends listed here for anything from analysis to microscopy to general support that I've received through this project. Thank you.
So great talk. I'm just going to jump in here. I have a really quick question.
Yeah.
So would you consider combining both your molecular and your morphological data sets together and then estimating the phylogeny that way instead of doing it separately?
I think that's a good idea. That's a method I haven't ever done before, so I would have to learn about it. But I think it would make a lot of sense to do that.
Yeah, I know there's different programs. Like I've used Mr. Bayes in the past. I think you can do it in Beast as well. But I think it would be something really interesting to look into.
That makes sense. Thank you.
And we should have time for one more quick question. If your question's already been stolen. I have one quick one in terms of, right, so your Pisarids come out non-monophyletic. There's a bunch of stuff that's falling in there. It looked like it included lycosids, big families. Are there characters now at that deeper node, so the one that actually does unite the Pisarids, that make sense and that then show, sort of support the inclusion of what's getting nested within?
Yes, I think so. I would have to do a little more analyzing to get several, but I know that one thing that all Pisarids have, including the ones that I think would be Dolomitidae, they all have this projection on the pedipalp called the distal tibial apophysis. So they all share that one, and then Dolomitidae has a few unique features.
And while the next set of slides gets put up, Jerica, go ahead. There's one more question in the chat. Other ecological or behavioral traits as opposed to morphological?
Now, that's a great question and not something I've gotten to look into yet. I haven't gotten the opportunity to observe these spiders much in the wild, but I mean, that's also something I'd love to add and look into.
Hey, thank you very much.
Thank you.
And with that, we are on to Jerica Jameson, who will talk about bat evolution.
Jerica, I think you need to unmute.
Unmuted now, sorry. Yes, I am super excited to share the work I've been doing in my PhD, looking at the convergence of glucose and fructose metabolism across nectar and fruit eating bats. Now, for most people, when they think of bats, they think of these little cartoons down in the corner, but bats are actually an extremely diverse group of mammals. first emerged right after the KT boundary 64 million years ago. And they've been evolving and diversifying ever since, putting them at currently 1,474 species. That means over 20% of all mammals on the planet are bats. And what makes this group really great for studying diet and dietary guilds are they have evolved pretty much every dietary guild you can think of. So here I've split the bat phylogeny into its suborders, Yangochiroptera and Enterochiroptera. You can kind of think of the Yangochiroptera as the tiny bats over in the Americas, whereas the Enterochiroptera are the larger species in Africa, Asia, and Australia. Not a perfect dichotomy, but it works for this presentation. And what's really fantastic about the evolution of nectar feeding and fruit feeding specifically is that it's happened multiple times in bats. So we have an origin of fruit feeding in the Yangochiroptera, an origin in the Enterochiroptera, and then multiple evolutions of nectar feeding from each of these fruit feeding evolutions. And one big caveat is I'm going to be saying nectar feeding, fruit feeding a lot. These bats do eat other things besides nectar and fruit. However, these are the main substances that compose the diet. So nectar feeding in particular has been a really classic example of morphological convergence in bats. Nectar feeding bats have repeatedly evolved elongated rostrums, very long tongues, and reduced dentition to help them feed on their floral resources. However, this doesn't necessarily happen in other bat species who lack these characters, but also eat some flowers in their diet. So a big question I wanted to ask in my research is metabolism of simple sugars changing convergently as we see with morphology, or is there more flexibility at the biochemical level across these independent evolutions of high sugar diets? So glucose and fructose are the main components of nectar, and they're monosaccharides, as we probably all remember from biochemistry class. And the way I look at their metabolism is through respirometry. I will feed either a solution of glucose or fructose that is labeled with carbon-13, which is the heavier isotope of carbon as opposed to the common carbon-12. What's really great, though, is regardless of where a molecule comes from or how it is processed in between consumption and use it for energy, in the mitochondria, everything is oxidized to CO2. And because I feed this isotopically labeled simple sugar to the bats, that isotopic signal is preserved in the carbon dioxide produced by the mitochondria. I can then use the ratio of carbon-13 carbon dioxide to carbon-12 carbon dioxide to approximate how much of the sugar I just fed the bat is being used for energy. What this actually looked like in practice is me feeding a bat and then placing them in metabolic chambers for about an hour and a half. At set time points, I sample the air containing that CO2 and store it in a vial for later. After a good field season, I'll have a couple hundred of these that I can then bring back to Canada to process on our stable isotope analyzer. I have been incredibly fortunate to be able to sample bat species in 6 different countries across the world that represent both of the independent evolutions of fruit feeding, 5 independent evolutions of specialized nectar feeding, and 3 lineages where they have some kind of facultative nectivory. What this data looks like is these kind of curves where time is on the x-axis and the proportion of metabolism being fueled by my sugar meal is on the y-axis. However, when you have 24 species, it's easier to break them down into PC axes for visualization. So for glucose oxidation, PC1 here is really showing just the magnitude of the oxidation capable by the species. So very low oxidation is over here in cluster D, whereas the high oxidation is in cluster C. And then the change between the 30-minute time point and the 60-minute time point is shown on PC2. For fructose oxidation, we have a very similar story. cluster H has very low oxidation overall, whereas clusters E, F, and G have very high oxidation. And cluster G here is showing the oxidation level at 10 minutes, whereas E is showing the oxidation level at 30 minutes. So what was really amazing with this data is that when we look at these two groups, when we look at oxidation of these two sugars, is we can actually map dietary guilt very easily onto this data. So these low oxidation groups are entirely composed of insect feeding bats that have very low levels of simple sugars in their diets. Then all of the fruit bats are also clustering together. But when we get to the nectar feeding bats, we have two different groupings here, which is not what is traditionally thought of when we think of bat dietary evolution. Usually we think bats started out as an insectivorous group, then they evolved frugivory, and then they evolved nectivory. But nectivory seems to be more of a mosaic than we've been giving it credit for. And having these two groups of nectivory brought up another question. So we know that the bats are progressing from insectivory to frugivory. That's a simple change in the metabolism in this PC space. But when they're moving to nectivory, can they move from the frugivorous phenotype to either nectivorous phenotype? Or after that move from insectivory to frugivory, do they have to move to this intermediate, more centralized phenotype before moving on to this more extreme phenotype? So to do this, I had to kind of hodgepodge together a phylogeny because most bat phylogenies are done at the family level. So for the root of my tree and the first couple of nodes, I used the Alvarez Carretero phylogeny of mammals and just pulled the bats out. And again, that was really just to get the origin of bats and then the relationship of the fruit-eating guilds to their insectivorous neighbors. Then for my two fruit-eating families, I substituted the Alvarez Carretero topology with the topology of family-level trees that are thought to have more accurate branching patterns at the genus and species level. Now, all of these trees were developed with different molecular markers to give branch lengths. So I just took the topology from the consensus topology from these three trees, and I used cytochrome B sequences to generate branch lengths for my sampled phylogeny. So once I had my tree, I was then able to test whether this stepwise pattern of dietary guild evolution is present in metabolism, or this free pattern where after developing fruit feeding, you can move to either nectar feeding guild phenotype. So I did this by generating stochastic character maps on trees in PhyTools, and specifically I restricted the character matrices so that only transitions for either shown here for either the stepwise or the free model were possible. And then I used MVMorph to simulate a multivariate Brownian motion model or a multivariate multi-peak Ornstein-Uhlenbeck model for my first two PCs for both glucose and fructose for both the stepwise and free pattern. And the result, that was just to see if the phylogenetic signal was explaining my data or if there were selective pressures going on. And for each of my conditions, the Ornstein-Uhlenbeck model was much preferred. So then I compared all of these Ornstein-Uhlenbeck models for glucose and fructose between the stepwise and the free pattern to see what better explained the data. And what I found for glucose was that the free model was slightly preferred. However, it didn't reach statistical significance. But for fructose, the stepwise pattern was significantly preferred by a large margin for explaining how the metabolism of the sugar is evolving. And if we look at a representative oxidation graph for each of my clusters for glucose and fructose, these results kind of make sense. So when we're moving from insectivory to frugivory for glucose, we're really just developing the ability to oxidize the sugar at all. And then when we move from frugivory to the central nectar feeding clade, we actually see a decrease in the overall amount of glucose oxidation. And we think that has to do with insulin insensitivity that we see in this group. And then we also see the more extreme group has this increase in glucose oxidation. So it kind of makes sense you don't want to decrease oxidation just to then increase it. For fructose, however, we see the same increase in overall oxidation, but then that central nectar feeding clade is represented by a shift from a 30-minute maximum oxidation time point to a 10-minute maximum oxidation time point. And that final shift to the more extreme nectar phenotype is just increasing that oxidation at 10 minutes. So if we take all of this back to my original question, are bats metabolizing sugar similarly across independent evolutions of nectar feeding and fruit feeding? Yes. For fruit bats, there's a clear convergence in how they metabolize both glucose and fructose, which shows frugivory is exhibiting consistent selective pressures on both of these sugars when they're metabolized. For nectar bats, it's a little more complicated. So they are converging into two different groups. So nectivory is applying different selective pressures to glucose and fructose metabolism, which taken a step further means nectivory is not one unified guild, but is actually more of a patchwork of selective regimes. So we can imagine a scenario where we have a bat that has a more central group phenotype for glucose, but a more extreme group for phenotype for fructose. We could imagine the opposite, so an extreme phenotype for glucose, but a central pattern for fructose, or two central groupings or two extreme groupings. And in fact, we do see bats that exhibit all of these different pairings. And so this is really the first time we're able to look at nectivory not as this monolith, but as this diverse group of selective regimes that we've previously thought of as just eating flowers. Final thank you to everyone who made this giant undertaking of collecting data possible. And I am slightly over time, but thank you so much for listening.
Okay, great. And not so much overtime that we don't have time for questions. We can do one quick question while the next person gets set up.
There's a question in the Q&A.
Yeah, so they asked if there's any bats that transition to nectar feeding that revert back to feeding on insects. And no, there is a fruit feeding transition that then reverted, but those are fairly rare bats and that are hard to catch, so I wasn't able to include any data on them.
There's also a quick question about whether the sampling was done in the wild.
Sampling was done in the wild. I converted rental cars into mobile labs.
Okay, thank you very much. And we will get going in just a minute with Ethan Briggs and our second spider talk of the session.
Just checking that everyone can hear me.
Yep.
Oh, awesome. All right. So, all right, guys. So, yeah, thanks so much to everyone for coming to my talk today. I'm going to be telling you about my PhD research on the biogeography and the systematics of Australian tarantulas. Oops. So as a tradition in Australia, I'd like to acknowledge and sincerely thank the traditional owners of the lands that I worked on across the beautiful Australian continent. Now, we're just going to dive straight into it. We're going to introduce you all to tarantulas. So in case you're not familiar, tarantulas are known for being these really large, these really hairy spiders, and which is also given this quite scary appearance when a tarantula gets threatened, as in the photo on the right. But I'm here to tell you the tarantulas are not all bad. As you can see, my overall excitement when I got to hold my first American tarantula. And then there's some absolute beauties out there, like the Brazilian jewel tarantula. Now, hopefully I can convince you that tarantulas are an evolutionary biology stream. You know, they're a super diverse group. We've got 1,100 named species, and they have a lot of different interesting ecologies. Like we have arboreal species and we have cave dwelling species. And they're also quite taxonomically challenging. We have examples of conserved morphologies, especially in closely related species. And then we've also got examples of convergent morphology in distant related species of tarantula. Additionally, they're really biogeographically interesting. So from this map here, you can see that tarantulas have done a good job at making their way across almost the entire world. But this is kind of counterintuitive because tarantulas also are considered to have this low propensity for dispersal. So we have these stationary females that typically live in a burrow or retreat for their entire lives unless disturbed. And then we've got these low dispersing offspring, which have been only known to disperse via walking and not been mediated by aerial assisted dispersal as seen in other spider groups. And particularly the last 5-6 years or so, tarantula evolution has become this global interest, and we've seen this progression of studies which have been either incorporating more and more taxa into their sampling or by using bigger and bigger data sets. And oftentimes this has taxonomic consequences. So specifically for tarantulas, we had to erect, these studies led to the erection of several new subfamilies, and in lots of other cases, many genera are found to be not monophyletic. And of those studies that are interested in biogeography, the biogeographic origins of tarantulas is consistent with a Gondwanan origin, or meaning that tarantulas are likely present when all the southern continents were still connected to form that supercontinent. However, all of these studies are missing a major piece of tarantula evolution, which is the Australian fauna, which is, a whole major continent that we're really missing out on. And I can't really blame these researchers because there's pretty limited knowledge on these giant spiders, which is kind of crazy to me. So for example, there's only 2 ecology studies on any Australian tarantulas, and they're both on this same species here, Selenocosmia sterlingi. But from my early PhD research, we did find that the taxonomy doesn't necessarily agree with their biodiversity. And with currently, we have 6 named species, I was able to find there's at least 20. And given that the Australian fauna weren't sampled in these broader phylogenies, we haven't been able to test their higher taxonomic placement. So if the taxonomic placement is consistent with their morphological classification, they should be placed in the Selenocosmini subfamily, which is characterized by the stridulatory organ, and which allows tarantulas of this subfamily to produce an audible hissing noise. And the map on the right is the current distribution of the Selenocosmini subfamily, which is comprised of approximately 100 species, all in Southeast Asia and Australia. So because the Australian fauna are thought to be closely related to the Southeast Asian fauna, researchers have suggested that maybe they arrived into Australia relatively recently in the last 30 million years. But some people have doubted that because there's not been a continuous dry land connecting Southeast Asia and Australia. So this has led some researchers to argue that the Australian tarantulas are probably also Gondwanan, like the other groups of tarantulas. So here is where I sort of break my talk up into the sort of 3 key results I would like to talk about, which are the testing the taxonomic placement of the Australian tarantulas, testing their biogeographic origins, and as a bit of a cliffhanger, a little unexpected but really cool finding that we found. So in order to test if the Australian tarantulas are accurately placed in their high taxonomy, we need to start off with good old sampling. So this comprised of doing field work as well as visiting lots of museums around Australia. And the goal was to maximize geographic coverage and identify any unrecognized species that we didn't find from our previous study. And on this map here, we've got 2 colored circles. So the gray circles represent museum records, and the yellow circles are individuals I was able to sequence at least a 16S mitochondrial marker for. So as you can see, we've got a pretty good overlap in the distribution that we were able to sample. And in total, that ended up having 369 tarantulas collected from across Australia. And in case anyone is wondering what collecting tarantulas is like in Australia, I'm pretty sure these 3 photos sum it up quite nicely. So from that 16S data set, I made a preliminary analysis, and unfortunately, we were not able to recover a lot of phylogenetic signal. So hopefully you can see on this phylogeny, this is a maximum likelihood phylogeny, and the black circles indicate supported nodes. And the blue diamond on the phylogeny is the crown of the Australian group, So as you can see, most of the internal nodes are unresolved, so we're really not sure about the relationships within the Australian fauna. So to incorporate the Australian fauna into the publicly available transcriptomes from other studies, I selected 20 species that span the phylogenetic diversity to help try and resolve the relationships within the Australian fauna, as well as answer these bigger questions. I just chucked the bioinformatic pipeline in here in case anyone was interested. But something I wanted to point out, which was really important to me, was to try and test multiple data occupancy matrices to see if the amount of missing data affected the resulting topologies. And so we used 50, 60, 70, 80, and 90% occupancy matrices, and we ran them through IQ trees and Astral, as well as report the gene concordance factors. So to test that first question, are they accurately placed in their higher classification, i.e. in the Solenocosmini subfamily? I was able to reduce maximum likelihood tree from 82 all the way up to 700 loci, depending on data matrix. And we were able to confirm for the first time, based on molecular data, that the Australian tarantulas are indeed accurately placed. And what I want to point out is the Australian fauna are deeply nested inside of the Solenocosmini, with these other 3 species being found across Southeast Asia, being Indonesia, Singapore, and India. And data occupancy does have a little bit of an effect on the topology, but is mostly stable. So one pointing out here is in the 90% data occupancy matrix, we did recover a alternative topology at the higher classification, but this alternative topology was not supported, so we don't think it's an issue. So moving on, we now that we have that tree, we can look at the testing the biogeographic origins. So to do that, I used BEAS, and I thought I should point out a key calibration point I used, which was the crown of the Australian fauna. Oh, sorry, the crown of tarantulas. So we used the oldest known tarantula fossil at 100 million years as our lower bounds, and we used the oldest known Mygalomorph fossil, which is the broader group that tarantulas belong to, as the upper bounds, while also testing a birth, death, and a Yule tree priors, again, just to see if these priors weren't affecting or driving our results. So ultimately, we found out the Australian tarantulas were probably not present on Gondwana before it broke up. We find the crown age of tarantulas is 8 to 18 million years, and the stem age is 14 to 35. And on the chronogram on the right, we have a blue ribbon that goes from the top to the bottom, and this represents the continuous breakup of the Gondwana into its continents. So when you take these two things together, the Australian fauna being deeply nested inside of largely Southeast Asian species and a relatively young age of diversification, we reject the idea that the Australian tarantulas are Gondwanan, but rather suggest that they came from Southeast Asia relatively recently into Australia. However, the really cool part about this is, again, this would have required overwater dispersal, which is really interesting given that we're working on spiders that typically live in holes underground and people don't come across them very often. So it appears that they were able to conquer this challenge all the same. Now, to get to the unexpected result that I promised everybody, we need to look inside of the Australian fauna clade. And what you might immediately see is we get a bit of a different story when we talk about really high support. So within the Australian fauna, we get really low node and branch support, and we have these really short internal nodes as well. And to sort of highlight this, amongst these 7 species here, we recovered 3 different topologies depending on which data occupancy matrix we used. So when you take these 3 things in combination, this leads us to suggest this is probably signatures of a rapid radiation. Now, a rapid radiation followed by the dispersal into Australia is not completely unheard of. There is a handful of studies that also find this phenomenon as well, such as the blind snakes and the Livistonia palms. And the question I've been asking myself, as well as many people have been asking me, is, was this radiation adaptive? And at this stage, we're not sure. That's because most of the Australian species are undescribed, with only 6 of them having names. So realistically, there's probably 30 to 40 species, I think would be a reasonable estimate, of course, depending on species concept. And this sort of highlights a need. We still need to do a major taxonomic revision of this group. So if you take nothing away from my talk, these are the 5 points I hope you'll remember me for, is that we added a major missing piece of tarantula evolution, that being the Australian fauna. We give an example of recent dispersal in a traditionally Gondwanan lineage. We give an example of overwater dispersal, or what is a dispersal poor taxon, which makes us to maybe have to reevaluate what these spiders are capable of. The importance of testing several data occupancy matrices, and of course, highlighting a novel rapid radiation that in the Australian tarantulas. So with that, I'd like to say thanks so much to my PhD supervisors, the labs I've been a part of and am a part of now, as well as the spider sampling team over my PhD, as well as all the organizations and societies that help fund my research. So with that, thank you so much. Here's my email if you would like to contact me later on. Thank you.
Okay, very nice. We have a first question in the chat already, which is that you've piqued Melissa Olive's curiosity about how they made it across the sea to Australia. Could it have been vectors or how? Speculate.
Oh, that's really cool. So at this stage, we're not really sure. That we're sort of leaning towards the idea that there is some possible island hopping. So we have seen in events where tarantulas have been caught on in floods, for example, been caught on like debris such as plants or trees and branches and things like that. But there's still, and so maybe island hopping through sort of short dispersal events over a long period of time. That's probably, I would say, our best guess.
Okay, and another question in the Q&A from Becky Chong. Great talk. I was curious if you had any idea about the historical biogeographic patterns that might help explain the recent introduction.
So there's a bunch of really cool studies, particularly on plants. So in the sort of last 30 to 40 million years, we see a lot of crossover in fossils, which sort of correlates to Australia potentially getting close enough to allow dispersal. And then you see it really ramp up in sort of the last 15 million years or so, which, you know, things like birds and other reptiles have been also. So I think it's purely driven by distance, getting closer and closer.
And we've got questions in the chat now from Sarah Morris. Great talk. Are these the only tarantulas who stridulate, or are there others?
Oh, that's a good question.
So
there is multiple subfamilies can stridulate, but they all have a different. The placement of their stridulatory organ is different. So in my group, they're close to the fangs, whereas in one of the African groups, they're closer to the coxa, or sort of almost, I guess, maybe the shoulder equivalent for tarantulas.
And then, okay, we've got time for a couple more. One question about the calibration. Was the root node the only one that you constrained, or were there other calibration?
Okay. That's an awesome question as well. So in our analysis, that was the only one we used because tarantulas don't have very many fossils. So there's only 3, and only one of them's 100 million years old. The rest of them are quite young, around 16-ish million, I think. So, and I didn't want to, I was a little bit nervous to use other secondary calibration points from other studies. So we just constrained it to that one calibration point.
Okay, and Karen, will you go ahead and get your slides set up? Final question. No tarantulas in Tasmania? Asks Karen.
No, we haven't found any yet.
Okay, why not?
We think it's because they dispersed in from the north. So tarantulas are sort of absent from the southern margins and the southeastern margins of the Australian continent. So that's probably why they just potentially haven't had enough time to get down there.
Great. And with that, we will move on to the last talk of this session. There's going to be a 30-minute break, but please come back for our second session after that. And another origins question by Karen Salazar.
Can you hear me okay? And see my presentation?
Yep.
Okay, the presentation is okay on my phone. Okay. So, hello everyone, I'm Karen Salazar, and I'm very happy to present my work in this symposium. And today I will tell you about the phylogenomics and evolution of Areopis ladybird beetles thanks to museomics as part of my thesis at the Sorbonne University and the Natural History Museum of Paris. South American Andes is a large chain of mountains localized in the west part of South America from Venezuela to Patagonia. And this cordillera has a very complex topography and is one of the main promoters of species diversification in the continent. There are different studies in the evolution of the biota of South America. Many Andean South American groups, as these examples, had origin in the Miocene or Miopleistocene, and most radiation in the Pliopleistocene. Origin area was the Central Andes for this group, and the diversification of this group was linked to the upland of the Andes and or the climatic oscillation during the Pliopleistocene. Most of these studied groups has a restricted distribution or are not endemic in South America. So the idea was to choose a group to explore this hypothesis and bring new ideas about the evolution of the South American biota. My model was the genus of ladybird beetle Areopis with 22 described species. All of them has a small size with similar coloration patterns. We can see some picture there. And this is an endemic group of South America, has a wide distribution and also on a large elevation range from 0 more than 5,000 meters in elevation. We can see in peak the distribution of Areopis. To answer this, if this promoter has an effect in the diversification of these beetles, previously I had to answer preliminary questions as, for example, what is the taxonomic diversity? What is the geographical distribution? What are the phylogenetic relations in this group? Because we don't know, or very few, about this information for this group. I need find material, become familiarized with the taxonomy, nomenclature, make a specific identification, identify the patterns of the geographical distribution. And for that, I study different natural history collection around the world because I don't have the possibility to do field work or get fresh material. So I found a lot of material in the collections, including the Thai material. And I did a deep taxonomic revision, doing a morphological exploration, obtain distribution from the labels, to better understand and identify these species. I use museomic approaches to get molecular information from this historical material. The samples comprise specimens from 186 years to 1 year old to collection data. I use a non-destructive protocol to DNA extraction, allowing to conserve the sample in the museums one more time. So I use a standard protocol to Illumina library preparation and use a shotgun sequencing method. And after the bioinformatic analysis, I recover many genes, mostly mitochondrial genomes. We can see how look like the mitogenome of Areopis. It's typical in most of insects. And also, I recover most repetitive genes in the nuclear genome. These DNA sequences were useful to do phylogenetic inference. I use 59 specimens of Areopis. I use the nuclear and mitochondrial genes. I use different inference phylogenetic methods, test the evolutionary models and skin partition. And after that, I recover Areopis as a monophyletic genus, closely related to other South American genera of ladybird beetles as Adalia and Cicloneda. Here I present you the first phylogenetic inference to this genus, and we can observe within the genus there are 4 main clades. After here I did a hard work for 3 years. So the result obtained in my thesis for this part helped to take some clues to answer the main question of my project. So we know now Areopis is more diverse than I expected in the beginning. We have now a better understanding about the taxonomy of this group. We know now more about the distribution pattern and the phylogenetic relationships thanks to museomic approach and museum specimens. So the objective of this part of my research were infer the origin and process of diversification of this group, determine the geographical distribution over evolutionary timescales, and identify and relate the possible cause of diversification in this group. Relating the observed distribution of the species in the phylogeny is evident the most then live in the Andes, and specifically in the Central Andes. A few ones live in the south of Andes or in the north, and other we live in the west and the east part, but also ones live in the Pacific Islands. Regarding the habitat, we can see most of these species live in highlands, and few ones or other ones live in the lowlands outside of Andes. I use two time calibration approaches, one with a large sample of 5 outgroups and 8 fossil calibrations. I use also a second calibration method based in secondary calibration, and I use the broad phylogeny of Coccinellidae. The origin of the common ancestor of Areopis was estimated around 9 million years ago in the Miocene, and the first diversification take place during the late Miocene and Pleistocene, and the major and rapid evolutionary diversification of this group occurred during the Plio-Pleistocene. In the same way, with the second calibrations, the origin of Areopis was estimated a little early, around 16 million years in the Miocene, and the rapid evolutionary and diversification also during the Plio-Pleistocene. Now, I combine geographical distribution of extant species and the dated phylogeny for ancestral area reconstruction. I divide South America in 10 areas. The analysis was performed in bioGeoVirs. I test the 6 models, and I choose the best according to the IC. So the biogeographical analysis with the best model support of the central area, ancestral area where Areopis was the Central Andes, and most lineage diversified inside and from the Central Andes. In the clade 1, we have fierce and early dispersion event to the Pacific Island, one Pacific Island. The Pacific coast of South America was colonized 4 times independently. From the Central Andes, one lineage colonized the mountains in the north central Cordillera, and another in the eastern Cordillera of Colombia. Colonization of the eastern part of South America occurred at least 5 times independently, and the most recent colonization events involve the eastern part and some Pacific islands, and occurred in the last 1 million years ago. So the biogeographical analysis show some keys to understand the evolution pattern of Areopis. Areopis originated when the Andes was formed, but elevation was lower than today. The most intense and recent periods of Andes upling occur between the 11 and 4 million years, especially in the central and north Andes. We have to consider the elevation of Andes was very complex and no homogeneous along all the Andes. For example, here we can see two close regions in the Central Andes had a different elevation at 16 million years, but after 5 million years, both has a similar elevation as today, around 4,000 meters. Then the formation of these mountains, especially in the Central Andes, split the population of Areopis, create barriers to dispersion of this population, and transport some lineage passively to high elevations. Reason why today most of the Areopis species are endemic of high elevation habitats in Andes. And the recent radiation of Areopis linked with the climate oscillation in the Pleistocene glacial and interglacial cycles create dynamic environment to facilitate both the colonization of the new areas, for example, the areas in low ecosystem, ecosystem in low elevation outside Andes, and isolation of the population in the specific habitats. The expansion of hyperactivity also in the western margin of South America has different outcomes from the Miocene to the Pleistocene, and we can also associate this hyperactivity expansion with the diversification of some lineage of Eriopis in the Pacific coast of South America. Marine integration in South America in the Miocene and the Pleistocene may explain why colonization of the S part of South America occurred only recently. Our results are consistent with many other studies. Eriopis is a predator of aphids. Aphids are closely related with the plants, so suggesting that their evolutionary story are correlated. In high altitudes of Andes, there are endemic and adapted alpine flora, as illustrated in these images. Today, these plants serve as a habitat to ladybird beetles and other arthropods. Consequently, these plants may act as a micro-refuge in the mountains where temperate species can be refugied in extreme environmental conditions in the past. Supporting this hypothesis, we observe that the diversification time of one family of these Andean plants overlap with the diversification times and distribution of Eriopis. Eriopis species today live in habitats with high elevation, cold and dry, others in median elevation, cold and humid, other ones in low elevation, dry and warm. So thermal resistant adaptation of these species may play an important role in the diversification and adaptation of these species. For example, in the Pacific coast, we have very extreme ecosystems, but also we have some plants persist and diversify in this kind of ecosystem. In the Pacific coast, most of the region is flat, but there are also small mountains with fog where some species of Eriopis live. So maybe this area, the small mountains, also serve as a refuge for this species. This was an integrative thesis work and involved different approaches, museumic approaches, museum specimens to answer evolutionary questions. We have pioneer information about the evolution story of this South American genus. Eriopis is a potential model, but we need completing most of information. The Andes was the ancestral area of the dispersal center for most of lineage of Eriopis. The diversification of Eriopis was probably mainly driving for most intense period of Andes upland and the climatic oscillation in the Pleistocene, and particular microhabitats in highland and the coast has acted as a micro-refuge for this species. An ecological adaptation made crucial during the evolution of this group. We have to complete gaps of information, taxonomic gaps, geographical gaps, to understand better the evolutionary story of this group. We have to test, for example, temperature elevation, oxygen tolerance, dispersion capacity. My thesis is not the end of this research. This is only the beginning. It opened the door to many other questions in evolution. Thank you so much for the attention. Thank you to my advisors and the labs where I did my thesis. Thank you so much.
Okay, great. We have time for a quick question or two. There's a question in the chat asking if there's from Daniel Schoenberger asking if there's any evidence of introgression resulting from the climate change.
Yes, I mentioned, but in my phylogeny, I have only the opportunity to include one specimen for a species. So in some case, we have doubts of about introgression in the species. So for the future, we have to include more specimens for a species and to continue to explore the taxonomy to include all the samples and all the species we identify. So this is only a beginning because we need to include many other samples to try to understand this evolutionary story.
Another question from the Q&A this is from Becky Chong. Nice work. She's wondering about the nuclear genes that you were able to get and how that compared in the older and the newer museum specimens.
Yes, I use most of the nuclear genes which I recover from my samples was the most repetitive genes from the nuclear genome, histones or ribosomal sequences. But also I get some genes with a few copies in the genome. Yes, for example, according to the age of the specimen, we have negative correlation. So for all these specimens, we obtain less nuclear genes. But for most of them, we have obtained the mitochondrial genes, but the nuclear gene was the most difficult. For example, for all the sample, the one of 186 years of age, we obtained few portion of the ribosomal genes in the nuclear genome.
Okay. Let's have a round of virtual applause for all of our presenters in this first of the Mayr sessions. Very stimulating. Enjoy your half-hour break, and remember that it's a different link that we tune back into in 30 minutes. Hope to see you all then.